Good morning, everyone. Today we are going to start our new topic, that is artificial methods of vegetative propagation. Earlier we studied about the vegetative propagation in natural methods, right? By natural methods, how it is propagated, we studied about that. There were three topics in that. There were stem, roots and leaves. Yes. But now we are going to start our new topic, that is artificial methods of vegetative propagation. How we can propagate artificially. So there are few methods in this. Can you see? Cutting, layering and grafting are the commonest methods of artificial propagation. Cutting, layering and grafting. As well as there is one more topic in this, that is tissue culture. Tissue culture. Tissue culture. It is tissue culture. Tissue culture. So this is layering method. This is cutting method. Let us start our new one. Let us start our new topic. That first is cutting method. Can you see this cutting? Yes. In this what happens? Stems are cut into pieces and planted into the moist soil, not in the barren soil, in the moist soil. Can you see this? This is the parent plant. Yes, this is the parent plant. From here, what happened? One stem is cutted. One stem is cut. Not cutted. One stem is cut. Then it is that stem is planted in the moist soil. Then what happens? It starts growing. Yes. So some examples are sugarcane, rose, and china rose. Is that clear? Let us begin with the next layering method. That is layering method. So in layering methods, what happens? You see, in layering, a branch is bent and covered with moist soil, while still a branch is bent and covered with moist soil while still attached to the parent. Can you see this? Can you see this picture? This is the parent plant. One stem. Can you see it is bent? One stem is bent, and that stem is still attached with the parent plant. Yes, it is still attached with the parent plant. Now, it is done in such a way that the growing tip remains above the soil. Growing tip. Can you see this growing tip? This is above the soil, right? It is above the soil. Then what happens? New roots arise from the branch is in contact with the soil. The piece of branch means the part of branch which is under the soil. There arises new roots. Can you see the new roots? Yes. Once rooted, the new plant is separated from the parent plant. When once rooted means it is rooted, the new plant means once it is completely rooted, means completely grown, it can be cut. So what happens? The new plant is separated from the parent plant. It can be cut and it can live an independent life now. Is it clear? So once again we will revise this layering method. In layering method, what happened? A branch is bent. What happens? A branch is bent. Yes, a branch is bent and covered with moist soil. Can you see? A branch is bent and is covered with moist soil, and it is still attached with the parent plant. Okay, is that clear? Now it is done in such a way that the growing tip, जो growing tip दिख रहा है ना आपको, it remains above the soil. ये soil के ऊपर रहेगा ताकि ये grow कर सके. Is that clear? Then new roots arise from the part of the branch under the soil. Yes. You can see this part of the branch which is under the soil, it has new roots in that. Then when it is completely rooted, means when it is completely grown, what can you do? You can cut it. You can cut or you can separate the daughter plant from the parent plant. Is that clear? This is layering method. What are the few examples of it? Few examples are cherry, jasmine and grapevine. Few examples are cherry. Jasmine and grapevine, as well as strawberries. as well as strawberries, as well as strawberries, as well as strawberries. Is that clear? Let's begin with the next, that is grafting. In grafting, you have to remember two parts especially. That is cayenne. Photo is in the next slide, shows the next page. The first is cayenne, the part with desired quality. Stalk, rooted base receiving the cayenne. Junction, vascular tissues used to allow growth. Now what happens over here, you see. In the next picture only, see, this is grafting. What happens? Grafting is a technique that joins two plants into one, combining characteristics of both plants. Is a technique that joins two plants, into one, joins two plants into one, two into one. Is that clear? Combining characteristics of both the parent. Is that clear? So what happens is cayenne stalk and junction is there. I will show with the help of the diagram. See, this cayenne. Cayenne means which you want to put. Cayenne means which you want to put. And stalk means where you want to put the cayenne. Is that clear? It is written here that cayenne means part with desired quality that you want to put. Is that clear? Suppose this is a half plant. Okay. What you want to put in this half plant, that is known as cayenne. Which you want to put. Want to put. Is that clear? Want to put. Then stalk, rooted base receiving the cayenne. Means where to attach. Means this cayenne. This is cayenne. Then where to attach this cayenne? In stalk. Is that clear? Any doubts? Cayenne means which you want to put. And stalk means where you want to put. Where you want to attach the cayenne. Is that clear? Junction means these two plants, vascular tissues, xylem and phloem. Yes, the xylem and phloem fuse together. What do? They fuse together. What happens? It's fused together. The xylem and phloem means the vascular bundles fuse together so that the growth of the plant can take place. Is that clear? Examples are, you can grow varieties of apples, pears and mangoes using this method. Is that clear, everyone? Yes. See, with the help of a diagram over here, it is cayenne which you want to put, and stalk, root stock or stalk, what you want, where you want to attach it, where you want to attach the cayenne. Is that clear? Graft union. Now it has been union. What happens? Labbed and wax. Is that clear? Nothing else. Then last topic is tissue. Last point in this topic is tissue culture. The another point in this topic is tissue culture. Is that clear? So what happens over here? New plants are grown in sterile test tubes. Sterile means very hygienic. Means 99.99% germs are not there. It is 99.99% clean because few, like, it is also a dust, right? Means soil is also a type of dirt only. Therefore, it is 99.99%. Is that clear? New plants are grown in sterile. Grown in sterile means very hygienic test tubes using a small tissue sample that is known as calyx. What happens over here? Suppose this in test tube it has been grown. Okay, it is grown in test tube. It is very hygienic. Okay. Then what happens? It is a soil over here. A small tissue. A very small tissue is placed in the soil. Is that clear? The very small tissue is placed in soil. This is known as calyx. Is that clear? Means a very small tissue. You can see this very small tissue. This very small tissue is grown in a very hygienic test tube, and then it in the nursery you can see this one. So the very small tissue is known as calyx. Okay. Let us erase it. Now it is thousands of free germ. thousands of germ-free plantains can be generated. Exactly, you see. What I am starting from detail. This is suppose a test tube. It is very hygienic. This is just soil, and a very small tissue. And a very small tissue is placed here. That is known as calyx. A very small tissue that is known as calyx. Exactly, a very small tissue is placed here. That is calyx. Is that clear? So it starts growing over here. You know, it will start. It will start growing. Yes, it will start growing. It will start growing. It will start growing. It will start growing. Is that clear? It will start growing. So thousands of germ-free planters can be generated using this method. It will start growing. So many of them are there. Can you see this picture? You see, many plants are there at once only. You see, many plants together. Yes. So thousands of germ-free planters can be generated. Thousands of germ-free. Because why germ-free? Because it is hygienic, very hygienic. Yes. Therefore, a germ-free planter can be generated. Examples means tissue culture is used in orchids and bananas. Orchids and bananas. You found this method usually in nurseries. You know, in nurseries they give you a test tube. Yes, they give you a test tube. In that test tube, there is a small tissue. It grows, grows, and grows. Yes. Then after a bit of growing, what can you do? You can grow it in a soil. You can grow it in a soil. Whatever it is suppose banana or what. You can grow it in a soil. You can grow it in a soil. Then it will grow. Is that clear? My diagram is very poor, so please don't see that for explanation. And yeah, I just do diagrams. Okay. So then it will start growing. Is that clear? So this is the end of this topic, that is artificial methods of vegetative propagation. Okay. So you know there are how many methods? There are four methods of vegetative propagation. Artificial vegetative propagation. That is, first is cutting. A healthy stem is cut and planted into the soil. Then it will grow. Layering. A a branch is bent and is still attached with the parent plant, and the what can I say? This growing tip is is in the top, is in the above, is above the soil, is present above the soil. Is that clear? The new roots arise where the branch is in contact with the soil. Yes, once rooted, you can cut that. Okay. The next is grafting. I always told you there are three terms or two terms you can remember. That is scion, stock, and junction. Scion means part with desired quality. Stock means scion means want to put, what you want to put. Scion means want to put. Okay, stock means where to attach the scion. Is that clear? Junction means fusion of both the vascular tissues. मीन्स यहाँ पर भी तुम्हारा जाइलम होगा, यहाँ पर भी फ्लोएम होगा, यहाँ पर जाइलम, यहाँ पर फ्लोएम. So both have to fuse together. Is that clear? Both have to fuse together. Is that clear? Both has to fuse together. Both has to fuse together. Is that clear? Both have to fuse together. Next, this is the photo of this. Next is tissue culture. I already explained that it is present in the test tube. The test tube is very hygienic so that it can be germ free. Okay, a small tissue is placed in the test tube which is known as calyx. This is calyx. See, this is calyx. Small tissue is known as calyx. Is that clear? Thousands of germ free planters can be generated used for orchids and bananas. Thank you for watching, everyone. In the next video, I will try to finish the sexual reproduction in flowers. Thank you.